Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR pyqs December 2023 Shift 2 question paper. This is part C questions that I am discussing. This is part 4 of the discussion. I will be dealing with cell biology, biochemistry, cell signaling, applied biology methodology, DNA replication, protein synthesis, genetics. Coming to the first question. Given below is a schematic representation of the tDNA region of a binary vector used for genetic transformation of plants. So this is the binary vector. It has got the left and the right border. And this is the promoter gene poly A, promoter gene poly A. The figure shows the presence of restriction sites for ECO-R1 and probes labeled 1 to 4. So this is the ECO-R5 region which is restricted, and the probes for southern hybridization. So 1, 3, 2, 4 are the probes that are used for southern hybridization to analyze copy number of tDNA in the transgenic plants. Given below are probes or combination of probes that are used by researchers for southern blotting following digestion of genomic DNA with ECO-R5. Which one of the following options represent the correct combination of probes that would identify single copy integration events from both flanks of tDNA? So this is ECO-R5 digestion. There are two spots, that is over here and well as here. So half of the probe should recognize this portion and the other probe should recognize this portion. So you should need two probes or a single probe, as in probe 2, which will identify both the ends, that is left border also as well as right border side also. Okay, we will see which two probes will identify. So probe 3 and probe 4, yes, that can be used. Probe 3 will give you this side banding, and probe 4 will give you this side banding. So 3 and 4 could be used. Probe 1 and 3, they are both on the same side, so it cannot be used. Probe 1 only, probe 1 only on one side, so that also cannot be used. Probe 2, yes, it will hybridize on both the sides, that is here also it will give you one band, here also it will give you another band. So probe 2 could be used. Probe 1 and 4 also could be used on either side of the cleavage site. So that is also correct. So which are the correct statements? Correct probes. A is correct. D is correct. E is correct. B is incorrect. C is also not correct. So A, D, E is the correct. A, D, E only. Then next one. Following statements are made regarding heterosis breeding in plants. We have to see for all correct statements. So heterosis is the superiority of F1 hybrid. It is observed only in the F1 generation, not in F2. A superiority of F1 hybrid over both its parents in terms of yield or some other characteristics. So heterosis is always lowest in a cross between two genetically diverse. No, it will be highest. So option statement A is incorrect. Cytoplasmic male sterility may be used in heterosis breeding to eliminate emasculation. That is correct. The A line cytoplasmic male sterile and B line used for maintenance of cytoplasmic male sterile line are isogenic lines differing only at a specific locus. That is also correct. Heterosis can be retained in the F2 generation. No. It is seen only in the F1 generation. So D is incorrect. Answer is 3, that is B and C only. Then next one. A knock-in cassette was constructed in order to introduce the beta-casein gene in an animal cell. So you want to introduce beta casein gene into an animal cell. So you do knock-in method. The viable clones were selected using double marker system, the G418 neomycin analog and ganciclovir. Identify the correct designated cassette from the options given below. SOH is sequence of homology, GO is gene of interest, NEO is neomycin resistance, and TK is thymidine kinase, that is for the ganciclovir. So what the things you have to take into consideration is that the positive that is neomycin should be between one selection, that is the positive selection marker. It should be present near the gene of interest. This is the gene of interest. This is the positive selection marker, and both should be flanked by sequence of homology. Whereas the ganciclovir, it is the negative selection marker. It should be present outside the sequence of homology, that is represented by TK, thymidine kinase. So here the question is identify the correctly designed cassette from the options given below. Option 1 is correct. Whereas option 2, if you see, both the markers are within the sequence of homology, so it is not correct. Here also third one, if you see, negative selection marker is within the sequence of homology, positive selection marker is outside. That is not correct. Here both the markers are outside the sequence of homology. That is also not correct. So the answer here is option 1. Then next one. Certain statements are made below regarding radioreceptor assay technique. Radioimmunoassay is also an example of radioreceptor assay technique where you will be using antibodies. So it is a competitive protein binding method. Yes, that is correct. It is a non-competitive. No, that is not correct. Radioligand is present in excess of unlabeled ligand. No. Radioligand is present in lesser amount than the unlabeled ligand. So C is not correct. Unlabeled ligand is present in excess of radiolabeled ligand. That is correct. Activated charcoal incubation and further centrifugation lead to separation of free ligand in the charcoal pellet. That is also correct. Activated charcoal incubation and further centrifugation lead to separation of bound ligand in the charcoal pellet. That is incorrect. Which of the following option is the combination of all correct statements? So A, D, E, that is option 3 is the answer. Now if you take a radioimmunoassay as an example, how does it work? This is the radioactive antigen bound to antibody. You add in lesser amount. So all the antibodies are filled with the radioactive antigen. Then you add unlabeled antigen, that is your blood sample or anything. So from there the radioactive antigen will be displaced by the unlabeled antigen. There will be competition between these two for binding of the binding site of the antibody. Then you precipitate the antigen antibody complexes with anti-immunoglobulin, that is another second antibody you add. So this is the radioactivity of precipitate, that is bound antigen, and this is radioactivity of supernatant. Then you measure the radioactivity and you will make a graph. From there you can calculate the unknown value of the unknown. That is what happens in radioimmunoassay. Then before coming to the next question, I will tell you regarding yeast to hybridization system. So it has got an yeast to hybridization system. It is used for protein-protein interaction studies. So it has got a reporter gene, a DNA binding domain and an activation domain. Only when the activation domain binds with the DNA binding domain, you can get the reporter gene being transcribed and translated. These two are transcription factors, this DNA binding domain and activation domain. Now the query protein of interest is fused with the DNA binding domain and it is known as the bait, and this is the activation domain where protein from the protein library, that is the prey, is fused with the activation domain. Now when the prey interacts with the bait, that is when both the proteins have got site for interacting with each other, that is bait interacts with the prey, the activation domain will bind to the reporter gene and there will be transcription and translation of the reporter gene. This is what happens in yeast to hybrid system. So here is what is taking place in yeast to hybrid system. Yeast to hybrid system allows detection of interacting proteins. In the following schematic, various components of yeast to hybrid system have been indicated by letters A to F. So here are the various components. You have to identify which is the bait, which is the activation domain, which is the binding domain, which is the prey, which is the reporter gene. So which one of the following options provides a correct match of A to F and components of the yeast to hybrid system? So E will be your UAS. F will be reporter gene. So that will cancel out 1 and 2. Now coming to D. DNA is binding domain. DNA binding domain. Gal4 binding domain. D is Gal4 binding domain. So binding domain, it will be attached by the bait C. So C will be bait. A is your activation domain. So Gal4 activation domain. And B will be your prey. So B is prey. So that gives you the answer as option 4. Then next one. Given below are two figures X and Y representing the segregation of molecular markers in 12 individuals of a mapping population. P1 and P2 represent the parents and F1 the hybrid. The following statements are made based on the above figures. So this is figure X, this is figure Y. So figure X, okay, now these are the codominant markers. X represent codominant markers. You can see both the bands in the F1 progeny. This is the F1 progeny. Both dominant and recessive bands can be seen, whereas in here, in the F1 progeny, only one band is observed. The recessive band of parent 2 is not observed, neither in the F1 progeny nor in the parent 2. So this is a dominant marker. Now the figure X represent profile of codominant marker, that is correct, in an F2 population. Okay. Figure Y represent profile of a codominant marker. No, it is a dominant marker. So A is correct, B is not correct. Figure Y represent profile of a dominant marker in the F2 population. That is correct. C is correct. Figure X represent profile of dominant marker. No, it represent codominant marker. So A and C are correct and your answer is option 4. Now next one. Given below is a DNA sequence. This is the DNA sequence. In the absence of any other affecting parameters such as length, TM, GC content, which one of the following combination of PCR primer sequence would be able to amplify the above fragment? So these are the various forward and reverse primers that they have given. So this is the DNA sequence. I have shown it in dash because only this much region is required for the preparation of the primer. This is the 5 prime 3 prime end of the DNA fragment. Here other sequences are there, I have not shown them. So this is the 5 prime 3 prime DNA sequence. This is the complementary strand of the DNA sequence, that is 3 prime to 5 prime. So one primer will be over here. So that is G, T, C, T, A, T, C, G. 5 prime to 3 prime, if you see, option 2 matches. G, T, C, T, A, T, C, G. So that is one primer, and this one will be complementary to the second strand of the DNA, this primer. And its complementary will be ATGCGA, that is seen in option 2, ATGCGATG. So the answer here is option 2. Now next one. Which one of the following intermediate enzymatic reactions would be most effective in facilitating ligation of a blunt-ended insert fragment with a vector digested with EcoR1 restriction enzyme? So this is the restriction site at which point at which EcoR1 digest its sequence GAA-TTC. So you have to find out intermediate enzymatic reaction would be most effective in facilitating ligation of a blunt-ended insert fragment with a vector digested with EcoR1 restriction. So this is the EcoR1. You get two fragments like this upon digestion. So the insert is blunt ended, so it will not fix here because this will produce, EcoR1 produces 5 prime overhangs, it produces sticky ends. So these sticky ends have to be converted to blunt ends so that it will ligate with the blunt ended insert. So what you have to do for that is, first you have to incubate with mung bean nuclease. Mung bean nuclease, this helps in removal of sticky ends of 5 prime overhangs and turns them into blunt ends. That is what mung bean nuclease do. Then these restriction digested fragments, they will contain a 5 prime terminal phosphate. The presence of terminal phosphate creates significant problem in the blunt end ligation because it results in self-ligation of the vector backbone. Now you have digested with vector with EcoR1. So 5 prime phosphate will be present over there, which is not required because it will result in vector self-ligation. That means insert will not be present in the vector. The vector itself will ligate and sit there. Insert will not be there in the vector. So in order to prevent that, what you do is you remove the phosphate present at the 5 prime termini of the vector by using shrimp alkaline phosphatase. So the answer here is option 1. Option 2, if you see treatment of insert is already blunt ended, so you need not have to treat with declino DNA polymerase. Then third one, treatment of vector with polynucleotide kinase. Kinase add phosphate group, so it will result in vector self-ligation. So 3 is not correct, and for polynucleotide kinase it doesn't create blunt ends, so that is also not correct. Treatment of insert with clean out, again you are treating insert which is already blunt ended. So 4 is also not correct. So the answer here is option 1. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you. I have already done previous year's CSIR question papers also. You can go through my channel playlist for the same.